Our presentation is on the chemistry of snake venom by Grace Terrio,
Alexa Fernandez.
Geneva Lindsley.
And Miranda Standing.
So snakes inhabit every country in the world except for Antarctica, New Zealand, and Ireland, which is those little X's on the map. And approximately 15% of all snakes are venomous, and this is categorized by 3 different families, which include the Atractocypidae, Elapidae, and the Viperidae families.
So what is snake venom? Snake venom is a fluid created by modified salivary glands in a venomous snake's mouth. The color of snake venom can range from clear to a yellow color, and it's produced in glands located behind their eyes. It's important that they're encased in glands because it allows for the venom to not be introduced into the snake's blood.
So there are 4 primary purposes for the use of snake venom. The first being the immobilization of a prey, and the immobilization occurs because most venom affects neuromuscular and hematologic systems. And an example of a neuromuscular system is the junction between nerves and muscles, which the venom disrupts. Hematologic systems includes blood function, which is again disrupted. The second use of snake venom is to kill a prey, and this differs from immobilization because a snake releases and delivers more venom to its prey. The third use is to aid the snake in digestion. Venom aids in digestion because of certain cytoxins it contains, which helps to target and destroy cells. And lastly, snake venom is released as a defense mechanism when a snake feels threatened by its surroundings. So in general, there are 3 main types of substances that the venom of snake falls into, and these 3 include cytotoxins, neurotoxins, and hemotoxins. And as mentioned previously, cytotoxins induce the death of cells through the damaging of the cell membrane, cell lysis or apoptosis. And the cytotoxins that exist in venom are varied and specific in the type of cells that they destroy. For example, cardiotoxins are specific to the death of heart cells. The second group of substances that a venom may possess is known as neurotoxins, which largely affects the nervous system, and this is where paralysis of a prey occurs. This is because the neurotoxins disrupt the signal between neurons, which can be done through a blockade or the reduced production of neurotransmitters. The last group of substance is known as a hemotoxin, which affects blood processes, and this results in the destruction of red blood cells, which affects a prey's ability to properly clot blood, and this reduced blood circulation is then lethal to a prey.
Snake venom is also made up of a variety of proteins. There are between 50 and 60 different types of proteins that make up snake venom. Venom also contains some elemental metals such as zinc, sulfur, and copper. The venom also varies based on the species, and the closer that the species are related, the more similar the venom and the makeup of the venom is. And there are usually 6 to 12 toxic enzymes found in snake venom, and those vary based on the different types of snakes.
Let's go over some of the common enzymes found in snake venom. One of them is proteolytic enzymes. These catalyze the breakdown of structural components of tissues and is found in all venomous species of snakes. The next one is hyaluronidase. This enhances the diffusion of venom by causing reactions that break down mucopolysaccharide links in connective tissues. And the photo on the right shows a depiction of a monomer of hyaluronidase, and it's done in a style similar to the line angle structures. A phospholipase, A2, a neurotoxin that disrupts biological membranes and can lead to permanent damage and respiratory failure. Another couple common enzymes in snake venom are L-amino acid oxidase, a catalyst for the oxidation reaction of a number of amino acids. On the right, you can see an image of an X-ray crystallography of a Malayan pit viper L-amino acid oxidase. And above it, you can see the reaction equation. Adenosine triphosphatase is also believed to be one of the central agents resulting in the shock of victims and immobilization of smaller prey. These are some of the common enzymes you can find in snake venom.
Is snake venom an acid or base? The answer is it's an acid, which means that during a litmus test, the red litmus paper stays red and the blue one turns red. The pH, of course, varies between species, but 2 examples are the Russell's viper with a pH of 5.8 and a cobra with a pH of 6.6. The acidity of the snake's venom widely contributes to how toxic the venom is. As for solubility, it's extremely soluble in water and glycerin. And with this high solubility comes a low isoelectric point. An isoelectric point is the pH when the charge of the protein in the venom is 0. It's also known that the snake venom has the highest solubility and lowest isoelectric point of any plasma protein. So why is snake venom chemistry important? Here are some statistics relating to snake bites. On average, 4.5 million people are bitten annually, 2.7 million have crippling injuries due to snake bites, and around 100,000 deaths occur annually. This number has the largest effect on poor rural farmers and children in India and sub-Saharan Africa, which is obvious because someone living in more rural areas are going to have a lot more exposure to snakes and have more opportunities to get bitten.
There are a few misconceptions that come along with snake bites that are widely known by a lot of people. One of the most common misconceptions is that sucking out venom helps take it out of the bloodstream. However, the amount of venom that actually is taken out by this method is insignificant, if any, and unhelpful. Some also believe that cutting out the venom will keep it from spreading. However, this can cause more harm to the person and can lead to an infection. If you ever get bitten by a snake, what you should do instead is to make sure to restrict the amount of movement that you have in the affected area. Keep your heart rate low and stay as calm as you possibly can, and get to a hospital as soon as possible so that they can inject an antivenom in you.
And so how was this antivenom created? Well, in India, they inject horses with a gradually increasing dose of snake venom. And normally this is a mix of a lot of different snakes, and then they harvest the horse plasma to create their antivenom. This process is banned in the US, so the US had to figure out a different way to do it. Recently, UC Irvine created a synthesized polymer nanogel that binds to several key protein toxins. By binding to these protein toxins, the polymers keep them from bursting the cell membranes and causing the widespread destruction you typically see with snake bites.
And despite the potentially lethal effects of snake venom, there have been advancements to utilize venom for good. An example of this include utilizing venom to treat epilepsy, asthma, and arthritis, just to name a few illnesses. This is because a few of the proteins found in snake venom actually aid in nerve and muscular processes. More specifically, it was found that the venom of the Russell's viper is a hemostatic agent, which means that it can aid in the prevention of bleeding, and this can be utilized through dilution in order to prevent internal bleeding.
A quote by Rumi that really encapsulates snake's venom and how it relates to life is, "Snake's poison is life to the snake. It is in relation to man that it means death."
Okay, that's all for our presentation on the chemistry of snake venom. We hope you enjoyed.